Hello friends, welcome to BMH Learning. In this video, we are going to discuss about FRAP assay. Introduction. FRAP stands for Ferric Reducing Antioxidant Power Assay. The assay uses colorimetric detection method. The sample to be used in this assay can be of plant origin, serum, plasma, and other biological fluids. FRAP is a type of quantitative assay. Talking about the sensitivity of this assay, this is around 0.2 millimolar. The assay provides a quick, sensitive, and easy way to measure the antioxidant capacity of a number of samples of biological origin. The significant feature of this assay is that it is high throughput, adaptable, and is capable of even detecting the antioxidant capacities as low as 0.2 millimolar Fe2 positive equivalents. Principle. The assay is used for the measurement of antioxidant potential of various samples. FRAP does this by reducing the ferric ions to ferrous ions through antioxidants present in the samples. Once the ferric ions are reduced, a blue color is developed, which is read colorimetrically at 700 nanometer. The determination of antioxidant potential of samples is done using a ferrous iron standard curve. Results are expressed as Fe2 positive equivalents, which are in micromolar, or you can call it as FRAP value. Requirements: phosphate buffer saline, or PBS. The buffer concentration is 0.2 molar. The pH is usually kept at 6.6, which can be adjusted with sodium hydroxide or HCl. 1% potassium ferricyanide. So, 1 gram of potassium ferricyanide is dissolved in distilled water. The volume is made to 100 ml with distilled water. 10% trichloroacetic acid, also called TCA. 10 gram of TCA is dissolved in distilled water. The volume is adjusted to 100 ml with distilled water. Be careful while handling this chemical because it can burn your skin. 0.1% ferric chloride. 0.1 gram of ferric chloride is dissolved in distilled water. As done with other reagents, we make up the volume to 100 ml with distilled water. Now, keep all the chemicals in airtight containers and store in dark conditions in order to prevent oxidation and other unnecessary activities. Procedure. Sample preparation. It is recommended that you should prepare several dilutions of your sample in order to ensure that the readings are well within the standard value range. It's also recommended to use only fresh samples for most accurate and reproducible assay. In FRAP assay, a variety of plant and fruit extracts, beverages, and also serum and plasma can be used as samples. The extractions from the fruit, vegetable, and plants can be done using various solvents like methanol, ethanol, or acetone. The extraction method depends on the type of specific sample being extracted. So, several plant extracts are taken in various concentrations. The volume of each sample with different concentration is made to 1 ml using the same solvent employed for the extraction of the sample. Then, in all the test tubes, 2.5 ml of PBS or phosphate buffer saline is added. Thoroughly mix the contents in each tube. Then, 2.5 ml of 1% potassium ferricyanide solution is added in all different concentration samples. After this, each reaction mixture is vortexed well using a vortex shaker. Then the samples are incubated at 50 degrees Celsius for around 20 minutes. Once the incubation time is over, 2.5 ml of 10% TCA or trichloroacetic acid is added in each sample. The test tubes are centrifuged at 3000 rpm for 10 minutes. Now, from these centrifuged samples, 2.5 ml from supernatant was collected in separate test tubes. This is done for all different concentrations. Then, to these new separate test tubes having 2.5 ml of supernatant, we add 2.5 ml of deionized water. After this, in the same new separate test tubes, we add 0.5 ml of ferric chloride. This would give us bluish color formation. And then take measurement at 700 nanometer. Now you have to keep one thing in your mind, that is, a sample having more concentration will show higher absorbance. The opposite is true. We also need to make a positive control. So for this, we can use a certain antioxidant molecule like ascorbic acid or butyrated hydroxytoluene. To make a positive control, we adjust the volume to 1 ml as done with the samples, and the same protocol is followed. That is, all other chemicals are added in exactly similar volumes. In the same manner, we make a negative control also. Instead of adding any sample, in this tube, we only put 1 ml distilled water, and other chemicals are added in similar volumes. Now, this is important. The assay we are talking about is just for comparison. So, we are not going to make standards as done in other biochemical assays. So, we don't need to draw a standard curve and then compare the sample's reading with values of standards. This video was just to let you know how can you check antioxidant activity of a specific plant extract by comparing it with commercially available antioxidants. This was all. Thanks for watching.